Now we'll hand over the floor to Dr. Helena Maier from the Pirbright Institute, who is going to talk to us about replication organelles, I believe, Helena. So you just try and share your screen and we'll just let you know that's working okay. Okay, that at least seems to be fine. The floor is yours.
Turn my video off just because it will probably help with the internet connection.
Okay.
So I'm still here, just hiding. Okay, so pointer on. Oh. Okay, so thanks for the invite to present today. Yeah, as Dalan said, I'm going to have a bit of a change of topic and tell you about one of the major focuses of my research group, coronavirus replication organelles, and a couple of different projects that we've been working on for the last couple of years, some progress we've been making. So replication organelles, all coronaviruses, like all positive strand RNA viruses, when they infect a host cell, trigger the rearrangement of host cell membranes to form what's called the replication organelle. That's this step down here in the replication cycle. Replication organelles are a specialized organelle. They support viral RNA synthesis, and they also shield viral RNA and replication intermediates from host cell detection. So they're a really critical stage in the replication cycle. As a result, over the last 10, 15 years or so, they've been studied quite a lot. So we now have information about the conserved replication organelle for all coronaviruses. So the first structure that everybody's probably familiar with is double membrane vesicles, which you can see here for the alpha, beta, and gamma coronaviruses. And the second structure is called double membrane spherules, which I identified several years ago now in IBV infected cells. So these are kind of pinched out vesicles that remain tethered to regions of either zipid-ER or convoluted membranes, depending on the virus. They have quite a complex structure by nature of the fact that they've got two membranes. It requires positive and negative curvature of the membrane to make the vesicle body and the neck. And you can also see that there's electron density that lines the interior of the spherule. We know now that double membrane vesicles are the site of viral RNA synthesis, and some work a few years ago has demonstrated that there's a protein pore that crosses both membranes of the DMV, and that allows newly synthesized RNA to exit the DMV into the cytoplasm. So although RNA synthesis takes place inside the DMV, double-stranded RNA and also the non-structural proteins have been found to localize using immuno-EM across both the DMVs and the convoluted membranes, or zipid-ER and spherules. And despite the fact that the function of spherules is entirely unknown. So there are lots of things that we still don't know about coronavirus replication organelles. As I've just said, the function of spherules is unknown. Although as they're quite a complex structure, we hypothesize that they must play an important role. They're conserved across all coronaviruses, and they would have quite a high energy burden to produce. That implies they must do something. And we suspect that there's a protein scaffold that supports the formation of these structures. We don't have a complete understanding of the viral and cellular proteins that are needed for formation and function of the complete replication organelle, although lots of proteins are being discovered all the time. And we don't know how DMVs form, particularly in infected cells. There's no clear picture around the organization of the different RNAs or even the proteins within the replication organelle. So genomic versus subgenomic, for example, positive versus negative RNAs, and that's something that we're quite interested in. And it would be good to get a clearer understanding of conserved mechanisms between coronaviruses. Because of their central role in the replication cycle, they're a really tempting target for antiviral control. So if we can understand whether there are conserved mechanisms across multiple coronaviruses, that will be really powerful. So in the first bit of work, I've got a really super PhD student in my group at the moment, Patrick, and he has been working split between Pirbright and Diamond to do some advanced imaging. So Patrick developed a fluorescent IBV where there's GFP fused to the N-terminus of the replicase gene, fused directly to NSP2 in the case of IBV. And that allows us to visualize replication organelles. Ooh, I appear to have some animation on there that I didn't know. The virus, although it does have slightly reduced overall peak titer of virus that's produced, the RNA synthesis over a 24-hour period is unaffected by having this GFP fusion, so we're quite happy to use this virus. Using confocal microscopy, Patrick has confirmed that the GFP localizes associated with double-stranded RNA, a marker for replication sites, but also more importantly with bromuridine labeled nascent viral RNA. So we can see that the red and the green signals co-localize quite nicely here. He took this one step further to do correlative light electron microscopy. So this allows you to image live cells using the confocal microscope, then process your cells for EM, and image those same cells overlaying the confocal images with the electron micrograph so that you can see what ultrastructure is associated with the fluorescent signal. So we can see here that this region of GFP NSP2 is associated with a small patch of double membrane vesicles, which you can see here, and this bit in the middle contains some spherules and zipid-ER. This also actually was really pleasing because it's the earliest time point post-infection that we've been able to detect replication organelles for IBV. So using this newly developed tool, Patrick has gone over to Diamond to make use of their cutting-edge imaging capabilities. So he's been working with Maria Harkialaki at B24 to use the cryosoft X-ray microscope. This allows us to image through the entire thickness of infected cells, which allows us to ask a variety of questions that is just not possible to ask with electron microscopy. So we want to understand what the complete replication organelle looks like. And also, because we're now looking through the whole thickness of the cell, we're hoping that we'll be able to detect intermediates in DMV formation, because we're no longer looking for a needle in a haystack. We've actually got kind of the whole haystack to look at, unlike in electron microscopy. So the workflow here is to plate cells onto EM grids, infect with the fluorescent microscope, plunge freeze, so we're looking at near-native state hydrated cells, and then perform a fluorescent screen to identify those infected cells on the grid. This grid is then put into the X-ray microscope, and you can see here a mosaic of the whole cell of images that Patrick's taken. And on the next slide, I'm going to show you a video of this section here. So to orientate you, we've got the nucleus here, and we've got the cell membrane coming down here, and this whole patch here is full of replication organelle. Just to give you a single slice, we've got these small circles here are the DMVs, and then the darker patches of membrane here, you can see there's kind of lines and little dots. That's the zipid-ER and spherules. So you can see that the DMVs and the zipid-ER and spherules are completely interspersed. From the video on the previous slide, you can see that the replication organelle fills the entire volume of this thickness, or the entire thickness of this bit of the cell. So it takes up a really massive volume in the cell at this time point. The next thing that we can pick out from this data is that there are obvious differences between the membranes that make up the DMVs and the zipid-ER. You can see that the DMVs are quite a pale grey, whereas the zipid-ER and spherules are really dark. So that shows that there's differences in the proteins that are present within those membranes. And Patrick is just starting the fairly monumental task of trying to do some segmentation on this data set, which may reveal intermediates and DMV formation. Trying to pick out specific information from this whole data set. There's just way too much information in there. So hopefully the segmentation will give us a bit more of a handle on that. Patrick's also been working with Dan Clare at EBIC to do some cryo-electron tomography. So here we're looking at a different scale of the replication organelles. So we're now looking at kind of the real protein structures. So we want to hopefully visualize pores in the DMV membranes, as has been seen for the beta coronaviruses. And also, more importantly, actually to look to see what the protein scaffold that supports the spherules is. If we can gain a better understanding there, it might be possible to kind of infer things about function of these vesicles, but also just start us off on another series of questions. So the same workflow at the beginning: cells plated, infected, plunge frozen, do the fluorescent screening. But then Patrick takes the grids for focused ion beam milling. So here you can see this kind of bulge going. Here is an infected cell. This is where the green signal is. So you can mark this out for milling, and you end up with a 150 nanometer thick lamella through the cell, which can then be put into the cryo-electron microscope, which is where we now get this absolutely stunning video of a region of spherules and zipper-DR. Hopefully you can appreciate from this, compared to the tomogram that I showed in the introduction slides, that the level of detail and the resolution is just massively improved. So we can get an awful lot more information than we had before. So we've now got the highest resolution images of IBV particles and replication organelles that we've had to date. Resolution of the spherules is below 5 nanometers. So if we look at some of the structures separately, I can kind of give you a bit of a rundown of the things that we have picked out so far. So we've got DMVs, which is great. So you can see the swirly RNA content within these DMVs. And here we've got a pore within the DMV membrane, which is fantastic. A DMV diameter is about 250 nanometers or 200 to 250 nanometers. So we would never get the full thickness of or a full vesicle within the lamella that we're imaging. But we can approximate that there must be about 5 to 10 pores per vesicle. And Patrick is doing some sub-volume averaging so that we will hopefully be able to compare the structure for our pores with those that have been imaged previously for MHV and SARS-CoV-2. We've also got some virus particles, which is really lovely to see. So you can see the lipid envelope and also the organized RNP content lining the viral envelope. You can see spikes depending on kind of where in the virus particle that you're looking at, but you can't see them there. But the bit that I get more excited about actually is the spherules. So the first really striking observation is that between the two membranes of the double membrane spherule, there is quite obvious striation. So you can see these stripes between the membrane. This also extends into the zipper-DR, so you can see those stripes in various bits in the zipper-DR, but you interestingly never see that in the double membrane vesicle membranes. So that backs up our earlier observation that there are differences in the composition of the membranes between the DMVs and the zipper-DR and the spherules, but also does raise some interesting questions around how DMVs are formed, because it implies that they're not just directly formed from the zipper-DR membrane. If we look at one spherule in detail, sorry, this might make your eyes go a bit funny, but there's not an easier way to show you. When you get to the very edge of the spherule, hopefully you can see that some kind of spots appear, which are protein extending into the cytoplasmic side of the spherular membrane. And also, as it gets to the edge, the protein starts to form a kind of spiral structure. And this is all showing that the striation is making up a lattice within the membrane, and the protein extends from the cytoplasmic face of the outside membrane into the luminal face of the inside membrane. We can take just some fairly basic measurements from slices of the tomogram, and the distance from the outside through to the inside of the two membranes is approximately 200 angstroms, and the space between the two membranes is about 80 angstroms. We know from our earlier work that if you express NSP4 from IBV on its own, that zippers together the ER membranes. And interestingly, if you take the alpha fold structure for NSP4, you have transmembrane domains at either end of the protein. The total distance here is 208 angstroms, and the distance between these domains here is about 80 angstroms. So it's really tempting to say that this protein here might be NSP4. Patrick is doing the sub-volume averaging so that we'll be able to get some information about the structure here, and then it will be possible to see whether this structure fits into the density that he generates through that analysis. But we are also doing, hopefully, some cryotomography of NSP4 expressing cells to see whether this same striation and these same structures can be seen in the regions of zipper-DR that we'll get. The next bit of information that we've picked out from this data set is that the spherules clearly contain a cargo. If you look through the kind of center slice of the spherules, you can see that there's this content. It appears to have some repeating structure. So there is a potential that this could be RNP. Being that we don't see RNA synthesis associated with spherules, we don't really know what the purpose of this RNP within the vesicle would be, but it will be quite interesting to look into that in more detail in the future. And then the last bit of information that we've picked out from this data set is that it appears that some of the inner membranes of the spherules might be sealed. So we have some vesicles here where you can clearly trace the inner membrane through from a region of zipid through the spherule and back out again. So these clearly have an open neck, whereas others you seem to have a sealed inner membrane and then the outer membrane making a neck. Interestingly, you do get some of these vesicles with a really long neck, which we'd never seen before either. In other vesicles, there's definitely a lack of clarity as to what is actually going on. So it's not 100% clear what the situation is with the necks. But again, Patrick's doing the data analysis, so the segmentation and the subvolume averaging to see whether we can get any more information out. And that is by no means a minor task. So that's going to take in a good few months to get that information. But overall, we're really pleased with this data set. It's giving us a huge amount more information than we had previously. So moving on very briefly to the next little project that I'm going to tell you about. This is being done by Connor Hayden, a postdoc in my group. And here we're trying to understand the viral proteins that are responsible for formation of replication organelle membranes. For the beta coronaviruses, it's been characterized that expression of NSP3 and 4 alone is sufficient to make DMVs. Here we have expression of an NSP3/4 cassette from SARS-CoV-2, and that makes some beautiful double membrane vesicles, and these even have a pore that have been imaged by cryotomography. Our earlier work had shown that co-expression of IBV, NSP3 and 4 from 2 separate plasmids didn't give us any sort of vesicles, it just gave us regions of zipid. But we wondered if that was just as a result of the expression route. So we made a comparable expression cassette for NSP3/4 and put that into cells, and note we still just get regions of paired ER membranes that form these massive patches and swirls within the infected cell, within the transfected cell. So it appears that for IBV, expression of NSP3 and 4 is not sufficient to make DMVs. So instead of adding more and more proteins to our transfection, we decided to take a slightly different approach to work out the minimal components for making the different replication organelle structures. So we have developed what we've called, for want of a better term, a replicon-based expression system. And here we are isolating the different regions of the genome to give us a pointer of where to look. So we've assembled the regions of the genome using the TAR yeast-based reverse genetic system, and each of these constructs are under the control of a T7 polymerase. We've put a GFP tag on the N-terminus of the polyprotein, so that's either NSP2 or NSP12, depending on the construct. So in our delta S, we've got the whole genome just lacking the S gene. We've then removed all of the structural and accessory genes, and we're left with just the complete replicase ORF1AB. In our next construct, we've isolated just ORF1A, so the cofactors and transmembrane proteins. And in the final construct, we've got just the enzymes, so ORF1B. All of these have been assembled into the yeast plasmid and then transfected into BSR T7 cells, which constitutively express the T7 polymerase, along with the N, and in the case of the ORF1B construct, NSP5 to allow for cleavage of these proteins. And this system we quite like because it allows us to look at proteins that are expressed from a polyprotein at the correct stoichiometry via a cytoplasmic mRNA, and it just more realistically reflects the situation that you would get in a naturally infected cell. So far we've managed to do some fluorescence microscopy with this. So to remind you what a GFP NSP2 infected cell would look like, you can see that you get these GFP puncta in the cytoplasm. With our delta S construct, you can see some nice puncta in the cytoplasm, and the same can be said for the ORF1ab and also ORF1a expressing cells, whereas there are no cytoplasmic puncta for the ORF1b expressing cells. So that is telling us so far that expression of ORF1a on its own is sufficient for replication organelle-like puncta within the cytoplasm. And the next step in this is to do some clem analysis to confirm what the membrane structures are that are associated with these puncta in the cytoplasm. This is proving somewhat tricky. However, we're hoping that we've got an approach that will work shortly. After we've done that, we'll then sequentially remove proteins from likely ORF1a so that we can work out which protein or which NSPs are required for DNV and sterol formation. Hopefully we can get each of them individually, and then we'll be able to tease apart function of those structures. So to summarize, we have confirmed that replication organelles take up a really significant volume of the cytoplasm of infected cells by 24 hours post-infection. And we're hoping that segmentation may reveal intermediates in DNV formation. We have visualized pores in the IBV DNV membranes. So we're hoping to compare those to the pores that are formed by other coronaviruses. We have the highest resolution images of spherules for any coronavirus to date. They've got a really complex architecture. So they've got that protein lattice that I showed you that crosses both membranes as well as an organized cargo. And Patrick's doing all the data analysis for the segmentation subvolume averaging so that we can get the most out of that data. We've confirmed that expression of NSP3/4 from IBV is not sufficient for DNV formation, unlike for the beta coronaviruses. And using our replicon-based expression system, we've confirmed that expression of 1A alone seems to provide the minimal components for replication organelle-like puncta in the cytoplasm. And we're just doing the clem, and we'll do some tomography of those cells so that we can visualize the replication organelle membranes or the membrane rearrangements associated. So finally, to thank the people who did most of this work. I've already mentioned Patrick and Connor, who've done a huge amount of work. We've also had some help from Nicole, Sarah, Pippa and Jenny, and Bioimaging, our collaborators at Diamond and Oxford. And yeah, thanks for listening.